The promoter region is present on the template strand of the DNA that is going to be transcribed and is a particular sequence of DNA nucleotides to which the enzyme RNA polymerase will bind. Just ahead of this promoter region will be the gene that we're going to be transcribing. So right before a gene, you have this little sequence of nucleotides called the promoter region, to which the RNA polymerase will bind and then begin transcribing that gene. This is what will give rise to the mRNA molecule. While this mRNA molecule is being synthesized, the RNA polymerase will add new nucleotides to the 3' end of the growing strand. These nucleotides are going to be activated nucleotides, meaning that they have the 2 additional phosphate groups attached to them. When these nucleotides are added, it's going to be in accordance with the rules of complementary base pairing. So the nucleotides that are added are going to be complementary to these exposed nucleotides on the template strand of the DNA. As we've mentioned, a nucleotide with adenine on the DNA will result in a nucleotide with uracil on the mRNA. Thymine on the DNA will result in adenine on the mRNA. Cytosine on the DNA will result in guanine on the mRNA, and guanine on the DNA will result in cytosine on the mRNA. Once the nucleotides align themselves according to complementary base pairing, hydrogen bonds will form between the bases, and it will hold them in place. In the meanwhile, the enzyme RNA polymerase will link the sugar of one nucleotide with the phosphate of the adjacent one, forming the sugar-phosphate backbone of the mRNA molecule by creating these covalent bonds here called phosphodiester bonds. This process will continue along the length of the template strand until the RNA polymerase gets to a certain triplet. This could be one of 3 different triplet codes. It could be ATT, it could be ACT, or it could be ATC. And when the RNA polymerase reaches any one of these 3 triplet codes, it will stop the process of transcription. This is why these triplet codes are called stop triplet codes. Once the RNA polymerase has reached the stop triplet code, it will detach itself from the molecule and the mRNA will stop elongating further.